And the last spiral bacteria we need to discuss is Borrelia. That genus is the most numerous by number of species included, and actually that bacteria is the largest in family of Spiracetaceae discussed in your program. It is good stained even by gram technique. It is gram negative. As you can see in the center of the slide, that which bacteria is easily identified. It is elongated, have wide open coils, maybe irregular shape, and again microaerophilic by type of respiration. And that bacteria accounts for 15 to almost 30 endoflagella, and bacteria is highly motile by twist and rotation movements. And bacteria is classified into 52 species, but usually they all are divided into two major groups. It is a group of bacteria which causing Lyme disease and a group of bacteria which is causing different types of relapsing fever. Uh, Species of Borrelia usually have a very large similarity. They are related one to each other very significantly and differs only by geographic area they are identified. So in the contrast with other spiral bacteria, Borrelia depends on blood-sucking insects and it can be transmitted only through vectors. It requires some intermediate host to be transmitted from one host to another host. And in some species, property to be transmitted through generations in tick population was identified and described. And actually, some bacteria can be transmitted from one generation of tick to other generation of tick without any host. One unique property of bacteria, it is ability to change genetic structure, change DNA very quickly, even within one episode of the disease. So once subject is infected, bacteria can cause several relapses because of changing of genetic structure. And the changing will lead to change in antigenic structure, and previous immune response will be much less effective or ineffective at all. So very often diseases have prolonged duration with relapses because of that ability to rearrange DNA. Again, I will show you that picture of endoflagella. It is an electron microscope photo. You can see those filaments, kind of tube in the structure of spiral cell. It is a filaments of endoflagella, and bacteria can contract or rotate or move like a corkscrew drive or like a twitching. Actually, besides gram stain, again, we have the same three methods to identify bacteria more detailed. It is a silver impregnation when in yellowish background, here it is even almost white, you can see dark brown spiral shaped bacteria. It's looking like some worm of different size, but usually they're elongated and have significant length, easy to identify. Dark field microscopy reflected light is going the objective, and you can see that glowing, shining white color bacteria. Again, that technique allows us to see a live bacteria, and you will see the unique mechanism of motility. And the most detailed, the most precise method is immunofluorescence. Here in the slide, in the center of the slide, you can see the picture, even different stains are used, and you can see bacteria of different colors, green, orange, and yellow. It means that you can have antibodies against different even spiral bacteria, and you can recognize species of bacteria, you can recognize serogroup or serotype of bacteria if it is required, and very easy to differentiate. Immunofluorescence using always antibody labeled with the dye, and they're precise and specific for one known species or serogroup or serotype of bacteria. So that technique allows us to put the diagnosis only by microscopy. Cultivation still is difficult, and Borrelia, as any spiral bacteria, have very long doubling time. Borrelia, similar to Leptospira and Treponema, multiplies once or twice in a day, and usually time required to obtain pure culture is extremely long for diagnosis of the disease, and cultural method is not used in identification of bacteria. But still, if you need to cultivate bacteria by any reason, we have that BSK medium, similar to leptospiral medium, which is named in honor of scientists who designed it, Barbara Stoner and Kelly. And nowadays we have several modifications of that culture medium. In the left picture, you can see native culture medium without any indicators. And colonies of bacteria are very small because microbe is slow growing. They are almost the same color as agar, and sometimes it's difficult to identify if colony is small. So modifications of BSK medium usually use some promoters or indicators and allows us to visualize colonies much uh, better. We have the chromagar, common row of culture medium, which is used as an indicator for specific species of bacteria, and you can see the colonies are colored. In that case, in green color, it can be under different color, but much easier to verify that it is really Borrelia growing culture medium. Again, for faster cultivation, we're using biological methods, inoculation in animals or in chick embryo, and some species of Borrelia can be cultivated even in ticks, and laboratories who deal with Borrelia in detailed manner, they're using ticks for multiplication of microbes. Here, bacteria is growing much faster and in bigger amount. And of course, you will easily obtain pure culture without any contamination of culture medium. So, as we said, bacteria can cause two groups of diseases. First one, more prevalent, is Lyme disease or Lyme borreliosis or just borreliosis. Bacteria can be transmitted through ixoid ticks. It is a hard-bodied tick, and usually rodents acting as the source of infection, maybe some other animals, especially if it is a dog who is walking in the forest, ticks can stick to them as well. So human very often can become infected during some trips in the forest, camping, hunting, or just walking in the parks with with massive area of trees or high grass. And initially, tick is looking like a bug. In the left part of the slide, you can see two photos. Ixoid ticks, also known as hard-bodied ticks, the whole body is covered with rigid layer of chitin, and usually ixoids, they have brown abdomen, light brown or dark brown, but they always brown. So when tick is stick to the skin of animal or human, in the upper right part of the slide, you can see the photo, that bluish gray bubble. It is a stick to the head of a girl tick, and some blood can go because ticks injecting enzymes to prevent blood clotting, to suck it easier. So after extraction, or when tick will suck enough amount of blood, it will fall down and will look like that bug in the bluish background. It is extracted tick with huge enlarged body, but still you can see that small brown head with eyes and some organelles in the front of the tick. So Lyme disease is characterized by three stages. First stage is acute stage. It is appearing immediately after tick bite or even during tick bite. As you can see, photos are numbered in the corresponding order. Initially, the tick will stick to the skin, or epithelium, and will start sucking blood. Red macular rash will appear, single element will appear around the tick, and it is due to injection of Borrelia in the soft tissues. So few days after tick is extracted, or it will fall down itself when it will suck up enough amount of blood, that lesion will remain here, but actually it will start growing in size, edges will be enlarged, and center will remain red. But mid-zone will become pale, skin will become intact, and with the time that lesion will grow and expand. Center will remain red, edges will remain red, but mid-zone will be pale normal color of the skin. And that thing will grow. It will expand, and very often that element is called expanding erythema, or erythema migrans, means it is migrating from the center to the edges. And in severe cases, it can occupy the whole parts of the body if several ticks will bite, and several lesions can be observed. And with the time, that erythema will just disappear, skin became pale and will take a normal color. So in that stage, besides local rashes, people can feel common symptoms of infection, weakness, loss of appetite, fever, pain in muscles, some headaches. But the most unique symptom is that erythema. If you will identify that lesion, you can put the diagnosis immediately and start therapy of the disease. But in some time period, that lesion will disappear, and actually bacteria will start migration into the bodies through bloodstream and some other organs. And disseminated infection, when bacteria will spread, it will manifest with neuropathy, lymphadenopathy, some pain in joints, and maybe arthritis. And in rare cases, it can lead to affection of meninges and heart, with some defects in conduction of signals in heart muscle. So disseminated infection is more difficult to recognize because actually symptoms are not very unique for that disease. Multiple diseases can manifest with pain in muscles and joints, and with some neurological disorders. And in that case, of course, confirmation by some testing method will be required. If disseminated infection will not be treated and recognized in properly manner, with the years it can transform to serious disorders. Usually, again, neural tissue is involved, and peripheral and neural tissue will suffer. And as you can see on the photo, it is acrodermatitis, means that innervation of regional tissues is affected, and skin will become thinner, layer will become smaller, loss of hairs will be observed in some years. You can see that skin became so thin that actually all blood vessels, even smaller, easily seen through the skin. Of course, skin will lose protective function. It will undergo chronic damages, cracks, and it can lead to bacterial superinfections. So as well, disease can lead to chronic arthritis, encephalopathy, and can lead to serious consequences during brain injury. So we had a vaccine against Lyme borreliosis, human vaccine. And nowadays, in almost every country, that vaccine is withdrawn by some reasons. And actually, the only measures of prevention of the disease are common measures to avoid tick exposure and any blood-sucking insect exposure, wearing specific protective clothes during camping or trip in forest, use of creams and some aerosols to prevent and to avoid tick exposure, and of course, check up of yourself and check up of your friends when you will come back home from forest or park trip. If you will see any insect which is still climbing by you, it's better to remove it and kill immediately. If any tick is already stuck in your skin, it's already injected, then you need to remove it by twisting movement, or we have specific tick extractors. And if you see that tick is mentioned by the description to Ixorid group, it's better to take the tick alive to sanitary station or to some specific institution who deals with Lyme disease, and give that tick for testing. If bacteria will be recognized, here it's better to take preventive course of antibiotics. If tick is healthy, then of course you will have nothing to afraid of. Second group of diseases is called relapsing fever, because the main symptom is fever, which will go in repeating episodes. And we have two major groups of relapsing fever. It is epidemic and endemic relapsing fever. Epidemic is identified only in humans and transmitted through louse. So very often it is called louse-borne relapsing fever, and the main species causing that disease is Borrelia recurrentis. As I told us from the name, recurrentis, recurrent means reappearance of the episodes. Second disease is disease transmitted through ticks, and usually it is animal disease. It's circulating in mammals and rodents, and human always is accidental host, but all symptoms are identical to epidemic fever. The only difference is way of transmission and the natural host. And in the group of tick-borne relapsing fever, we have a lot of different species, which are mainly named by the name of the tick, which is intermediate host for that Borrelia. All Borrelia are extremely similar, and actually they can be, and maybe they will be identified as a biotype of one Borrelia, but nowadays it is a still separate species. And different types of tick-borne relapsing fever are recognized in different geographic area, and usually countries, some precise countries, they have only one or few different species of Borrelia, and they are familiar with those diseases and those pathogens because of high accidents. But actually, again, management, therapy, and diagnosis of diseases are the same. So as we said, the main symptom of that disease, of that group of disease, is temperature, which is going in cycles. High temperature, low temperature, high temperature, low temperature. It is caused by mentioned already property of Borrelia, rearrange DNA structure and change antigenic structure in one host. So after Borrelia is injected into human or animal bodies, it will multiply and goes in the blood. And when a lot of bacteria will appear in the blood, subject or animal will have sudden onset of high fever up to 40 and even high degrees Celsius. And immunity, of course, will initiate protective functions, and a lot of Borrelia will be destroyed. It is gram-negative, so all properties of gram-negative bacteria are applied, and of course, release of endotoxin will lead to extremely high episode of the fever. In 3 to 5 days, our immunity will clear infection in blood and temperature will decrease. But actually, Borrelia is not present in blood, but it will hide in other tissues and start multiplication with rearrangement of DNA. And again, in some week or a little more, new serotype of bacteria will goes back into the blood and cause second episode of fever. And it can last up to months and half of the year till 10 episodes of relapses can be observed until bacteria will use all possible changes in DNA structure, and human will have antibodies against any possible serotype of bacteria. After that, usually disease subsides and convalescents are cured even without the treatment. Again, because of multiple different possible serotypes within every species, and we have a lot of species, actually vaccination is not available, but we have again the same measures of non-specific prevention, avoid tick exposure, and checkup of people in groups of risk to avoid the disease. Of course, if you see that subject is afebrile, but he will say that yes, few days ago it was extremely high fever. I was so bad I couldn't come to you. If the subject will come to you in afebrile period, then it's not the point to take a blood for diagnosis because bacteria is not here. But if your subject will come to you when fever is extremely high, it's not first episode. You need to take history of visiting some forest or park area, and if you suspect that it is maybe relapsing fever, especially if subject will say, yes, I was bitten by a tick or some insect, then you need to take blood in febrile period for bacteria, to look for bacteria. Diagnosis of disease again is the same as for any other. We have a ELISA test. Here it is shown a ELISA test for Lyme disease. Both IgG, IgM should be identified. Principle is the same. Micro-well plate, 96 wells covered with antigens of Borrelia recurrent or Borrelia burgdorferi is used to find antibodies in human blood. As far as both M and G classes are present here, it's not the matter which time period of disease is present in your subject. Just take blood, add in that set, perform required preparations. If color in the well will be changed, means test is positive, subject have antibodies, and you have your diagnosis. It is Lyme borreliosis. Test again can be performed usually in laboratories, required some several hours to obtain the result. And again, we have that rapid immune chromatography tests with the paper strip and uh, required reagents to identify presence of Borrelia burgdorferi itself, or we have some tests for antibodies against Borrelia. Principle is the same. You need to add sample obtained from the subject, usually it is serum, and apply it in that sample window. Add reagent which is supplied with the test strip, and if you will have positive result, so T test zone will be marked with the colored strip. C is a control, it always should be positive. And in nowadays, still the main idea is to prevent human from the disease by regular checkup and treatment of animals. And we have the same test system, and actually we have a vaccine for animals. Usually it is used for dogs, hunting dogs, or people who live in villages near a forest area. Dogs should be vaccinated to prevent infection of the host, of the owner, because usually dogs have very close contact with the people, and some ticks can bite dog and then bite human. And we have the same rapid diagnostic test, immunochromatography test for animals. Usually they are combined for multiple diseases such as filariasis and other. Principle is the same. As you can see here, it's not a strip, it is a dot, which will change the color in the case if disease is positive. So checkup of animals nowadays is the priority to prevent disease in humans. That's all for current lecture. We'll see you in other lectures. Bye bye.